Welcome to the video on transcription. So the overall goal of transcription is to start with a molecule of DNA and use it as a template to create a molecule of RNA. But before we dive into the details of this process, let's go ahead and talk about a few of the differences between DNA and RNA. Now notice both of these molecules, the abbreviated name ends in Na. That stands for nucleic acid. So you're both the same type of macromolecule, but obviously there's got to be a difference, right? The D and the R kind of give that away. So we're going to start with what those first letters mean. So DNA stands for deoxyribonucleic acid. The RNA is ribonucleic acid. And so the difference is the name of the sugar involved. So if you think back, remember, nucleic acids are made up of nucleotides, and each nucleotide has a phosphate group, a sugar, and one of the four bases, A, T, G, or C. The sugar in DNA is deoxyribose, whereas the sugar in RNA is ribose. Okay? Another difference between DNA is the number of strands. DNA is a double-stranded molecule, but RNA is a single-stranded molecule, so it only consists of one strand of RNA nucleotides. Now, the base that makes up those RNA nucleotides, there's a tiny difference there as well. Remember, in DNA, those bases were A, T, G, and C. But with RNA nucleotides, the bases are A, G, C, and U. There is no thymine in RNA. There's uracil instead. So if you're ever looking at a strand of nucleotides, like we will later, if you see a U in the sequence, that means we're looking at RNA. If you see a T in the sequence, you know you're looking at DNA. So the overall shape of DNA and RNA is also different. DNA is a helix, but RNA, it varies because as we'll talk about in 1 minute, there are 3 types of RNA molecules, and each one is going to have a different shape because it has a different function. Okay? So these are just a few of the differences between these 2 molecules. So let's talk more about what transcription is. And to really understand that, you need to know what it means to transcribe something. So when you're transcribing something, okay, you're kind of taking words from one form and putting them into another. Okay, so for instance, when I was in graduate school, one of my degrees was in education. And so a big part of my data were teacher interviews. So I would conduct those interviews in English words, but it was oral, right? I would speak them out loud. But when I went to publish my research, I couldn't just hand over all these recorded tapes, right? I had to sit down and I had to listen to my English words and I had to put it into typed English words. So it went from oral to typed. The language was still the same, but the format different, right? Oral to written. And so that's why we're calling this transcription, because it's a similar process. We're still speaking nucleotides, but we're going from DNA nucleotides, which have the deoxyribose sugar and A, T, G, and C. We're going instead now to the RNA nucleotides, which have ribose as the sugar and A, G, U, and C for the bases. Okay, so again, still the nucleotide language, but different, a little bit of differences there. Think of it as kind of like different accents of the same language. Okay, so during transcription, we're going to start with a double-stranded DNA molecule, and we're only going to use one of those strands as a template because we're going to make a single-stranded RNA molecule. So because RNA is single-stranded, we only need to use one of DNA strands as a guide. Now, the process of starting with DNA and making RNA is transcription. And technically, there are 3 different types of RNA that get made during this process: messenger RNA, transfer RNA, and ribosomal RNA. And those are what you see listed there. Now, even though all 3 types are made during transcription, we're going to pay the most attention to the messenger RNA, also called mRNA, because that is going to be the directions that we need to make proteins. Okay, and we'll talk a lot more about that later. But when we say that gene expression is DNA, then a molecule of RNA, and then we get proteins, the RNA we're really focusing on is the mRNA. Okay? Now, we need a key player. What enzyme is going to make this strand of RNA nucleotides? That key player is RNA polymerase. Normally, we call it RPOL for short. So this name should sound kind of familiar because think back to DNA replication. When our goal was to make strands of DNA nucleotides, it was DNA polymerase that did that. DNA polymerase made DNA. Well, now RNA polymerase is going to make the RNA. Okay? So where does transcription happen? Well, that's going to depend on the type of cell we're talking about. We tend to focus on bacteria in this class. And so what you have to ask yourself is, where's the DNA, right? Because that's what we're starting with when we're talking about transcription. We're going from DNA and we're making RNA. So where's the DNA in a bacterial cell? It's in the cytoplasm. It's just in that inside space, right? Now, if I wanted to get tricky and I said, where would transcription happen in a eukaryote? Well, that's different because where is DNA in a eukaryote? It's in the nucleus. So transcription has to happen where the starting material is. So that's the nucleus for eukaryotes, but it's the cytoplasm for prokaryotes. Because remember, bacteria are prokaryotes. All right, so there are actually 3 steps to transcription. The first is initiation, where we're kind of getting the party started. We have to get our key player to the start site. Then we have elongation, where we're actually elongating or building the piece of RNA. And then termination is where the process terminates. That means we're done. So where does transcription start? Well, it has to start at the beginning of a gene, right? That segment of DNA that codes for a protein. So the beginning of a gene has a sequence called the promoter. And the promoter is where RNA polymerase is going to bind to start the process of transcription. So we say the start site of transcription is the promoter. So during the first step of transcription, again, it's called initiation, RNA polymerase is going to bind to the promoter, which is just a sequence of nucleotides on the DNA molecule. At that point, because RNA polymerase is the only key player, we're ready for step 2, which is elongation. This is where RNA polymerase is going to travel down the piece of DNA. And as you can see in the picture, it's going to separate the 2 DNA strands. That's what you see here in blue is the DNA molecule. Do you see how a region of it is separated? And this is where the RNA molecule is being built. That's what you see in pink. Now notice something, y'all. Do you see this red arrow that's pointing towards the 3' end of the RNA? That means new RNA nucleotides are being added at the 3' end. So that means the 3' end is constantly getting longer. So we say that RNA is made, 5' to 3'. Okay, so that's going to be the directionality of all of our RNA molecules, 5' to 3'. So let's stop here for a second. Because RNA is also a nucleic acid, just like DNA, this strand of RNA molecules is going to be anti-parallel and complementary to the template DNA, right? Because that's a feature of nucleic acids. The strands are anti-parallel and complementary. So check this out. Do you see how the pink RNA molecule, right? It runs 5' on the left to 3' on the right. Well, look at this bottom blue strand of DNA, the one that's being used as a template. Notice it runs 3' to 5'. So the DNA template strand is anti-parallel to the RNA strand that's being made. Because again, on the end, on the right-hand end, the RNA is 5', the DNA is 3'. They're opposites, okay? And then if you look, the RNA strand that's being made is complementary, or base pairs, to the template DNA strand. So check this out. So notice, y'all, I'm looking here at the 3' end now because you can see the base pairing a little bit better. This G at the end of the RNA is pairing with this C on the DNA. This U on the RNA is pairing with that A on the DNA. Okay, so this is why I don't try to draw things actually curved like they happen, because it's sometimes you have to look at an angle to see the base pairing that is happening. Okay, but the sequence of the RNA is complementary to that template DNA strand. And we're going to show this on the next slide when we apply what you're learning. But notice, y'all, this 5' end that was made first of the RNA molecule, do you see how it's separate? It's hanging off of the DNA, and this region of DNA has reclosed, okay? What I'm trying to point out here is that even though these two strands of DNA are separate, it's only temporary. Once the RNA has been made for that region, the RNA lets go and allows the DNA to close back up. So that's what you see here on the left. This blue region, see how it's closed? That's because it's already been used to make the original piece of RNA up at the top. Okay, because you'll notice the 3 nucleotides up here on the RNA, A, U, G, are complementary to the 3 nucleotides down here, T, A, C. Okay? Now notice again, on the RNA, do you see how there's no T's, right? Because there are no thymines in RNA. So now during elongation, RNA polymerase is just going to keep trucking along down the DNA, adding nucleotides until it gets to the end of the gene. Once it hits the end of the gene, it says, okay, I'm done. And that means we're at the 3rd step, which is termination. And that's when the RNA polymerase is going to dissociate or let go from the piece of DNA. It's going to let go of the RNA molecule that it just made, and the process is finished. All right? So let's take a look at kind of what you would need to know on the exam, how I could ask you to apply this. So here's a piece of DNA. Notice it's double-stranded. The 2 strands are anti-parallel and they're complementary. We know we're looking at DNA because there's thymines in it. See all these T's? That's how you know you're looking at DNA. Now what I'm asking you is to give me the directionality and sequence of the RNA that will be made after transcription. So let's visualize this. So here's my double-stranded DNA. So remember, in order to make RNA, we need these strands to temporarily separate. So there they are. And now we need to use only one of these strands as a template because RNA is single-stranded. So we only need to make one strand of RNA nucleotides. So because RNA is always made 5' to 3', that means we need the anti-parallel DNA strand to serve as the template, which is the 3' to 5'. So here is the RNA molecule that's going to be made. Notice it runs 5' to 3', and notice the sequence is complementary to the DNA, okay? So notice here, y'all, when the DNA has an adenine, what does adenine pair with? Uracil, because we're making RNA, okay? When there's a thymine on the DNA, thymine always pairs with adenine, which RNA has, so that's good. If there's a guanine on the DNA, it's going to pair with cytosine, which RNA also has. Okay, so you just keep going through, but when you hit an adenine on the DNA, your brain may go, ooh, adenine pairs with thymine, which is true for DNA. We're making RNA, so you have to put a uracil instead, okay? So I put the answer down here. Here's our 5' to 3' piece of RNA. Again, you know it's RNA because of the uracils. So what I want to point out is this sequence is almost the same as this starting DNA sequence, right? It also runs 5' to 3', but if you swap out the U's with T's, notice they would be the same thing, okay? But because RNA doesn't have thymine, we just had to rewrite that sequence, but put U in place of T. All right, y'all, that is it for this video. Let me know if you have any questions.